Good morning, students. In this session we will know about the 11th standard biozoology, chapter number 8, excretion. The subtopic is mechanism of urine formation. In previous classes we already discussed what is excretion process, structure of kidney, structure of nephron. The excretion, it is a simple process, the process by which removal of nitrogenous waste product from the body is excretion. Here, what are the nitrogenous waste are formed in our body? There are three main nitrogenous waste are formed. One is urea, another one is uric acid, third one is ammonia. These three are the major nitrogenous compounds. Why it is a nitrogenous compounds? These are formed as a result of breakdown of amino acids. Breakdown of amino acid in the tissue. This breakdown of amino acid into urea. These are formed in the liver. So the liver is the principal organ for urine formation. So this takes place in the liver. So where the urea is formed, the question may be asked. The urea is formed in the liver by the process is ornithine cycle. Ornithine cycle. Ornithine cycle. Because ornithine is one of the compounds. So the ornithine is needed for formation of the urea. This ornithine cycle otherwise called as urea cycle. Urea cycle. It takes place in the liver. So listen, what this cycle is formed? Because this is the mitochondria. This is the mitochondria. Here, one of the amino acids is formed. This is glutamine. Glutamine. This amino acid is converted into urea. That means, ammonia. Glutamine is one of the amino acid is converted into urea. This ammonia. In the presence of carbamyl phosphate, here one of the enzymes takes place, carbamyl phosphate synthetase. Carbamyl phosphate synthetase. Again, the glutamine is amino acid to the ammonia, presence of carbamyl phosphate and ATP molecules and the carbon dioxide is this carbamyl phosphate is combined with the citrulline. Carbamyl phosphate, presence of citrulline, again it is formed citrulline. The same thing is a citrulline. Citrulline is converted into arginosuccinate or arginosuccinic acid. Again, the arginosuccinate is converted into arginine. Arginosuccinate is converted into arginine and the removal of the fumarate. Removal of fumarate. Again, arginine is converted into ornithine. Between the ornithine, formation of arginine into ornithine, here the urea is formed. The urea is removed and formed. So, I repeat this process, ornithine cycle. In all tissues, due to the process of deamination, that is the removal of amino acid, is converted into ammonia. Ammonia in the presence of carbamyl phosphate synthetase to form carbamyl phosphate. Carbamyl phosphate is combined with the citrulline to form arginosuccinate or arginosuccinic acid. Arginosuccinate is converted into arginine. Arginine again to form ornithine and removal of the urea. So ornithine is essential for formation of the citrulline. Then the arginosuccinic acid and removal of the fumarates. So this cycle is ornithine cycle. Ornithine cycle. What it says? The ornithine cycle explained to synthesis of the urea in the liver. Okay. Next, come to the process of urine formation. The mechanism of urine formation involves, there are three processes. How many processes? Three processes. One is glomerular filtration. Second one is tubular reabsorption. Third one is tubular secretion. Suppose the question may be asked, how many processes involved during the urine formation? There are three processes. What are they? Glomerular filtration, tubular reabsorption and tubular secretion. So main thing for filtration, reabsorption and secretion. I repeat, filtration, reabsorption and secretion. Now, how it takes place? The first process, that is glomerular filtration. So, normally, kidney receives blood or blood enters into the kidney by the main arteries, renal artery. You know, renal means kidney. Kidneys received blood by the renal artery. Once it enters into kidney, that means nephron, it forms afferent arteriole. Once it enters the afferent arteriole, it forms glomerulus. This is several glomerulus structure is formed. So the kidney receives blood from the renal artery into the glomerulus through the apparent arterioles. One more arteriole is efferent arterioles. Efferent arteriole. Here, why it is called glomerular filtration? This is the first step of the urine formation, which is formed in the glomerulus. Hence it is called as glomerular filtration. What is glomerular filtration? Filtration takes place in the glomerulus is called glomerular filtration. Mostly that fluid leaves from the glomerulus into the Bowman's capsule is called glomerular filtrate. Listen. Fluid, certain amount of fluid leaves from the glomerulus into the Bowman's capsule. This is called as glomerular filtrates. Here, apparent arteriole, another one is efferent arteriole. This is called as glomerulus. Glomerulus are covered by double-walled sac is called Bowman's capsule. These two structures are combined form is Malpighian tube. Malpighian capsule or tube or renal capsule. Here, the blood is mostly composed of larger quantities of the water, plasma protein, that means colloidal protein. Colloidal means large molecules of the proteins, amino acid, glucose and other nitrogenous waste products. How it is possible of the blood filter here? Due to the two forces occur. One is the hydrostatic pressure formed in the glomerulus. So the heavy pressure is under the pressure is formed here. The glomerulus are the large surface area which have the glomerular membrane. This is the glomerular membrane. This glomerular membrane have many pores. Through these pores, the blood and other substances except the blood cells and the plasma protein enters from the glomerulus into the Bowman's capsule. These minute pores allow the very small molecules, not larger molecules. So it allows only less than 5 nanometer smaller molecules. It does not allow the colloidal molecules because the larger molecules, because they have minute pores are there. Here, blood enters faster with greater force in the afferent arterioles. What is the meaning of afferent arterioles? The vessel, the blood vessel, receive blood from others through the renal artery, from the renal artery, is called the afferent arterioles. The afferent arteriole means what? This is the blood vessel smaller than artery. Arterioles. Once it enter into the renal artery, enter into the glomerulus, it forms afferent arteriole. Afferent arteriole again divides into many capillaries. These capillaries are called the glomerulus. The glomerulus are otherwise called the duct of blood capillaries. Duct of blood capillaries. This is the glomerulus. Here, the blood moves faster with greater force in the afferent arterioles, and it leaves from the glomerulus into out of Bowman's capsule through the afferent arteriole is slower. It is slower. So why it is nature, the afferent arterioles are narrower, the afferent arteriole are the wider. What is the difference between afferent arterioles? The size of the afferent arterioles is larger, so the blood flows faster with greater force, but the blood leaves from the glomerulus through the afferent arteriole is slower. Here, the afferent arteriole is narrower, the afferent arteriole is wider, so the blood flows very fast because the lumen, the space of the afferent arteriole is more than the afferent arterioles. So the blood is entered here. So the kidney produces approximately the glomerular filtrate is 180 liters in 24 hours. 180 liters, 170 to 180 liters. But not this all glomerular filtrate is enter into the other parts or enter. So mostly they are reabsorbed. Here in the reabsorption, glomerular filtration occurs by the three processes. One process, this process is hydrostatic pressure. Hydrostatic pressure. The first pressure. The hydrostatic pressure means fluid or liquid pressure in the glomerulus. This is the hydrostatic pressure. The hydrostatic pressure always may be 55 millimeter mercury, mmHg. This hydrostatic pressure always greater than oppositing pressure. Oppositing pressure created by the plasma proteins, by the plasma protein. This is the first pressure. This pressure is opposite by plasma protein in the capillaries. Plasma protein, albumin, is one kind of plasma protein, globulin. So these are the plasma protein opposite the hydrostatic pressure. There are two pressure is formed against the hydrostatic pressure. The first pressure is blood colloidal blood colloidal osmotic pressure. Blood colloidal osmotic pressure. This is the second type of pressure. Blood colloidal osmotic pressure. Colloidal means what? Larger molecules. Larger molecules. So osmotic means what? It also includes this water. This pressure is about 30 millimeter mercury. 30 millimeter mercury. What is another pressure is formed here? Here the Bowman's capsule. The fluid is present in the Bowman's capsule. This pressure is capsular pressure. Capsular hydrostatic pressure. Capsular hydrostatic pressure. This is about 15 millimeter mercury. 15 millimeter mercury. Listen carefully. Hydrostatic pressure is always greater than opposing pressure. Hydrostatic pressure is 55 mmHg. This opposite pressure, blood colloidal osmotic pressure is about 30 millimeter mercury. Another capsular hydrostatic pressure, the capsular pressure is 15 millimeter mercury. This two opposing pressure is produced by plasma protein in the capillaries. Listen here. What is net filtration force or net filtration rate? Listen carefully. The net filtration. What is net filtration? Here the hydrostatic pressure is 55 mmHg. This pressure, that is 55 hydrostatic pressure, minus 30 mmHg. 30 means what? Colloidal osmotic pressure. This is the colloidal osmotic pressure is 30 plus 15 mmHg. The 15 mmHg is the capsular pressure. Is formed in the fluid in the Bowman's capsule, which is about 15. This pressure is what? Colloidal osmotic pressure. This pressure is hydrostatic pressure. The remaining 15 is what? Capsular hydrostatic pressure is 15. What is the calculation here? The capsular pressure. So 55 minus 30 plus 15, that is 45. Here 35 plus 15 is 45. What is the remaining amount? 55 minus 45 is equal to, that is 10 mmHg. This 10 mmHg is called net filtration rate. So this hydrostatic pressure opposed by two pressure that are created by plasma protein in the capillaries, that is blood colloidal osmotic pressure is 30 plus capsular pressure is 15. 30 plus 15 is 45. So that hydrostatic pressure is greater than opposing pressure. 55 minus 45 is equal to, that is 10 millimeter mercury. This 10 millimeter mercury is net filtration rate. Okay, this is called as net filtration rate. This effective pressure, which is 10 millimeter mercury, is called ultrafiltration. The glomerular filtration, otherwise called ultrafiltration. So what is called GFR? This is the one more question. Glomerular filtration rate. Glomerular filtration rate. Here the volume of filtrates formed in the Bowman's capsule per minute is called glomerular filtration rate. The volume of filtrate formed per minute is glomerular filtration rate. In adult, the glomerular filtration rate is 120 to 125 liters per minute. Per minute. So here the volume of filtrate is formed per minute is glomerular filtration rate. In adult, which is about 120 to 125 milliliter per minute. So this is the GFR, that is glomerular filtration rate. Here it is glomerular filtration rate. So the approximately 170 to 180 liters of glomerular filtrate is formed in 24 hours. But urine output is only 1.5 liters. What about the remaining amount that are reabsorbed in second process? Here, what is the difference between the plasma? The glomerular filtrate rate is equal to blood plasma. The glomerular filtrate is equal to blood plasma. You know what is blood plasma? The liquid portion or fluid part of the blood is blood plasma. So what are the compound is present in the glomerular filtrate? Same to the blood plasma. That is except plasma proteins. So listen carefully. The glomerular filtrate do not having plasma protein like albumin. But plasma protein is present in the plasma. But that plasma protein is absent in the glomerular filtrate. Okay, so what is the concentration? Certain concentration of the glomerular filtrate isn't here. So one tabulation is given in our book. One is the substances. Another one is concentration of substances in blood plasma. So these are measured, it is milliliter. The second, another column is concentration of glomerular filtrates. So this filtrate, what are the substances are normally there? That is water. Another one is proteins. Next one is amino acids. Another one is glucose. The fifth substance is urea. Sixth substance is uric acid. And the seventh one is creatinine. And the eighth one is inorganic compounds. Inorganic substances such as sodium, chloride and potassium. So what is the amount of water is present in blood plasma? These are the 900, the same in the glomerular filtrates. What is this amount is present here? Just variation, it is 80.0, this is only 0.05. The amount of protein in blood plasma is 80. The amount of proteins in the glomerular filtrate is 0.05. What is this amino acid is present? 0.5. The same in the glomerular filtrate. What is the amount of glucose is present? That is 1. It is another one is 1. What is the amount of urea is present? That is 0.3. Here also 0.3. What is the amount of uric acid and creatinine? The uric acid is present 0.04. The same in the glomerular filtrate is 0.04. What is the amount of creatinine? 0.01. And in the same in the 0.01. What is the amount of inorganic substances? That is 7.2. The same in the 7.2. Okay, this tabulation. So, listen here, this amount of water similar, same in the blood plasma and the glomerular filtrate. This all the compounds, this amount of compound is similar to, that is same in the blood plasma and the glomerular filtrates. So, what is the difference, main differences between the plasma and the glomerular filtrate? Plasma and the filtrate are same, except the plasma protein is not present in the glomerular filtrate, but it is present in the plasma. Listen. The second step of the urine formation is tubular reabsorption. It is the second step. So nearly I explained that is 170 to 180 liters of the glandular filtrate is pumped per day, but urine is released only 1.5 liters per day. So nearly 99% of the glandular filtrate are reabsorbed into the body. The remaining substance are act as the waste. This must be eliminated from the body. This is called tubular reabsorption. So the tubular reabsorption that takes place in the nephron, particularly the tubules of the nephron or uriniferous tubules of the nephron. Proximal convoluted tubule, another one is Henle's loop, distal convoluted tubule and collecting ducts. So the tubular reabsorption takes place in the PCT, that is proximal convoluted tubule. Another one is Henle's loop. Third one is DCT, distal convoluted tubule, and fourth one is collecting duct. So the epithelial cells of the tubules of PCT, Henle's loop, DCT, and the collecting ducts reabsorb the useful substances, either active transport or passive transport, or it may be diffusion or osmotic process. What are the four important processes? Active transport, another one is passive transport, next one is osmosis, then the diffusion process. What is active transport? The movement of substances from the region of lower concentration to higher concentration requiring energy. What is the passive transport? Movement of substances from the higher to lower concentration do not require energy. The osmotic process, it is a reabsorption of the water. And the diffusion, it is one of the passive process. Movement of the substances from the region of higher to the lower concentration. Listen here, this takes place in the first part is PCT. That is proximal convoluted tubule. You know already this diagram. So this is apparent arterioles. Next one is apparent arteriole. This is the Bowman's capsule. Here, this is called as Malpighian tubule. Malpighian tubule next continues to form proximal convoluted tubule. This is a PCT. Proximal convoluted tubule. Proximal convoluted tubule again to form Henle's loop. Next Henle's loop is continues to form distal convoluted tubule. Distal convoluted tubule again connected with the collecting ducts. This is the collecting duct. So listen here, the first reabsorption takes place in the PCT, that is proximal convoluted tubule. So what are the substances that takes place here? One is water, next one is amino acid, next glucose, next is sodium, another one is urea and uric acid. So these compounds are reabsorbed in the epithelial cells of the PCT, that is proximal convoluted tubule. Because the wall of the proximal convoluted tubule easily absorb these compounds. Is most of the water is reabsorbed, amino acid, glucose, sodium, urea and uric acid. Small amount of urea and uric acid is reabsorbed here from the glandular filtrate. It is a fluid. Here the sodium is reabsorbed by the sodium reabsorbed by the active transport through the sodium potassium pump, exchange pump. Here the sodium is moving and the potassium is released here. So that is exchange of the sodium and the potassium pump. Listen here, one more question. How it is the sodium in proximal convoluted tubule is reabsorbed? Sodium reabsorbed by the active transport through the sodium potassium exchange pump. Exchanging of the sodium and the potassium in the PCT, proximal convoluted tubule. Okay, the water is reabsorbed in the PCT, especially water permeable channels in the PCT is aquaporins. Aquaporins are nothing but water permeable channels. This permeability means what? The wall of the tubules are easily absorb the substances or allowing the substances. Okay. Next, PCT is followed by Henle's loop or loop of the Henle's. Henle's loop. This Henle's loop are more permeable to water. More permeable to water and impermeable to sodium and the chloride. Listen carefully. The thin descending limb of the Henle's loop are more permeable to, is permeable to water, impermeable to sodium and chloride. So once the water is moved from the glomerular filtrate into the tissue fluid, so the concentration of the sodium and chloride is increased because this filtrate containing what? Water with the sodium and chloride. Once the water is lost from the glomerular filtrate, the amount of the potassium, amount of the sodium and chloride is increased. Once the urine passes from the thin descending limb to the thick ascending limbs, here the permeable to the thick ascending limb of the Henle's loop, permeable to sodium chloride and impermeable to water. Listen, these two regions. Descending limb are permeable to water, impermeable to sodium and chloride. Permeable means what? Water only allowing, but sodium and chloride is not allowing here. So impermeable of the sodium and chloride, but in the case of ascending limb of the Henle's loop, the sodium chloride is permeable and water is impermeable. Water is impermeable. So once the urine is passes from the ascending limb, this limb is called a descending limb. Another one is ascending limb. Ascending limbs. Once it enter into the DCT, that is distal convoluted tubules. Why the name is distal convoluted tubule? Distal means what? It is located just is the distance of the glomerulus. Proximal means what? Near to the glomerulus. It is a distal means what? Distant to the glomerulus. Convoluted means folded. Folded. Here, what it is reabsorb is it secrete, the wall of the DCT secrete potassium. Tell what is it? Potassium is important. So once the hypotonic fluid reaches into the, because the sodium and chloride is reabsorbed, the amount of water is increased. Once the water with the urine is entered into the DCT, increase the amount of water, immediately they secrete potassium and also passes urea and other salt in the DCT. Here, the absorption of the distal convoluted tubule is important because it is depends upon the body needs. Body needs. The absorption of the sodium and other substances in the distal convoluted tubules are regulated by hormones. What is the specific hormones? Is aldosterone hormone. Aldosterone hormone. You know, aldosterone hormone are secreted by the kidneys, particularly that is adrenal glands. So aldosterone, cortisone and aldosterone are secreted by adrenal cortex. So these mainly is involved to reabsorption of the sodium in the distal convoluted tubule. This wall of the distal convoluted tubule are secrete potassium. Potassium into the filtrates. Potassium into the filtrates. So this also absorb another one substance is bicarbonate, HCO3. Bicarbonate are reabsorbed in the wall of distal convoluted tubule which maintain the blood pH. What is the pH? The concentration of hydrogen and bicarbonate ions. So that is pH range is maintained by reabsorption of the bicarbonate in the distal convoluted tubules, and also the homeostasis of the potassium and sodium is maintained, is regulated in the DCT, that is distal convoluted tubules. So the reabsorption in the DCT and the collecting tubules responsible for are regulated by the hormones. Okay. Next, the reabsorption that takes place in the collecting ducts. Collecting ducts. Here, the water is reabsorbed. Water is reabsorbed. Once the urine moves from the DCT to the collecting duct, the urine once more become hypertonic. Because the more concentration, water is removed, the concentration of the salt is increased in the collecting ducts. This collecting duct is useful for is formation of concentrated hypertonic urine. So what is it? Our urine is what? Hypertonic urine. Hypertonic urine means that is more amount of the solid or salt in the urine. What is hypotonic? Hypotonic means more amount of the water. But before that, the fluid or urine become what is it? Hypotonic. Once it enters into the collecting duct, it becomes hypertonic. Hypertonic means the concentration of the solid is more than the water. So this region, this medulla region of the kidney, which is useful for production of or formation of concentrated urine. So that is tubular reabsorption. Tubular reabsorption takes place in the PCT, that means proximal convoluted tubule, Henle's loop, distal convoluted tubule, and the collecting ducts. Okay. What is the third process is tubular secretion. Tubular secretion is the third step in the mechanism of urine formation. So here, what are the substances are secreted? The wall of the tubules, particularly the DCT and the collecting ducts. The DCT, proximal convoluted tubules, and the collecting ducts. They secrete the three important ions or compounds. One is hydrogen, next one is potassium, next one is ammonia. These three compounds. And also it produces creatinine and other inorganic compounds. These all the inorganic compounds is moved from the peritubular capillaries into the filtrates. Listen here. This is what is peritubular capillaries? This is the apparent arteriole. Once the apparent arterioles moves, it divides into many smaller capillaries. The many smaller capillaries runs along with the other tubules of the nephron. It may be connected with the PCT, Henle's loop, and distal convoluted tubules. This is called a peritubular capillary. What is peritubular? Peri means what? Around the tubules. It comes near to the proximal convoluted tubule and the distal convoluted tubule. So the exchange of the substances from the peritubular capillaries, because they contain blood. Because blood moves from the glomerulus into the apparent arteriole. Apparent arteriole forms peritubular capillaries. These are the smaller capillaries around the tubules. Here, these substances, hydrogen, potassium, and ammonia and other compounds, move from the peritubular capillaries into the filtrates. Into the tubular fluid. The fluid present inside the tubules. Here, the most amount of the water is reabsorbed in the PCT, that is proximal convoluted tubule. Nearly 80% of the water is reabsorbed in the proximal convoluted tubules. Here, the Henle's loop is the main role for reabsorption of the water and impermeable of the sodium and the chloride. So, these changes that occur in the Henle's loop of the nephron, and the distal convoluted tubule also produces, that is potassium and reabsorption of the bicarbonates. Here, for hypotonic, urine moves from the ascending limb of the Henle's loop into the distal convoluted tubule and it reaches the collecting tubule. It becomes what is it? Hypertonic urine. Here, every hydrogen is secreted. So directly it moves from the peritubular capillaries into the filtrates to maintain the pH of the blood, body fluids. Here, the hydrogen is secreted. It is always combined with HCO3, HPO3 and the NH3. Here it is a bicarbonate, metaphosphoric acid. This is ammonia. So once the hydrogen is produced, every hydrogen is produced, are secreted into the tubules. It is form of bicarbonate. Another one is phosphoric acid, metaphosphoric acid. Next one is ammonia. Ammonia. So which maintain the pH of the body fluids. So these compounds always fixed as H2CO3. Next one is H2PO, PO4, and another one is NH4. NH4. So here it is the carbonic acid, phosphoric acid, another one is ammonium salt. Here it is what is it? Bicarbonate, metaphosphoric acid, ammonia, but it is always the hydrogen is fixed in the form of carbonic acid, phosphoric acid. Next one is ammonium salt or ammonium ions. The ionized form of the ammonia is NH4. NH4. The hydrogen is fixed as this like. Here, the every hydrogen is secreted in different segment of the nephrons. This hydrogen is secreted into the tubules and enter into the filtrates, and reabsorption of the sodium is occur. The hydrogen, potassium are secreted into the tubules and enter into the filtrate. At the same time, the sodium is reabsorbed. This will stop the reabsorption of the hydrogen from the filtrates. Hydrogen is secreted but not reabsorbed, but sodium is reabsorbed, sodium chloride, other things. So this is called tubular secretion. Why it is called a tubular secretion? So some certain substances are secreted in the tubules. It may be PCT, DCT, this part of the nephrons. Okay, the three processes or three steps are completed. Now, that is mechanism of urine formation. There are three processes: glomerular filtration, tubular reabsorption, and the tubular secretion. Okay, so finally, after the useful substance are reabsorbed into the blood and finally is eliminated as urine. So the urine is collected from the collecting duct into the, that is duct of Bellini, and into the ureters and eliminated. So this is mechanism of urine formation. This question is important question in excretion. So various questions are coming from this chapter, this lesson. So this video, I think it is useful to you, and observe and watch carefully and read well. Thank you.